Next speaker is David Ponder. David is from the University of Oklahoma. His talk is investigating the cell wall changes during grass lateral root emergence. David.
Thank you, sir. One of the reasons we were interested in lateral root emergence is lateral roots are important in both water and nutrient use efficiency in plants, and the other reason was that they are emerging from the interior of the root, and they have what seems to be a cell separation event as the lateral root emerges. So we were interested in the cell wall changes that occur during the lateral root emergence by using bulk cell wall analysis, and we wanted to identify the genes that regulate this the cell wall changes during lateral root emergence using gene expression. Now this is a little difficult to do because, as this picture here shows, this lateral root emergence is asynchronous, so they emerge at different points in time during the development of the plant, and cell wall material cannot be amplified like genetic material can be. So the way we have to go about this is a synchronous lateral root system, which here on the left is the auxin signaling pathway we believe is happening during lateral root emergence, and we can selectively inhibit that pathway using an auxin transport inhibitor, and then release the inhibition using exogenous auxin and get a synchronous lateral root emergence. And so here I've done some tests, and on the top you can see the number of initiated lateral roots that they have. Blue is a untreated control with no inhibition or exogenous auxin. Green is our treatment system, and you can see that it is much higher in the number of initiated lateral roots. And then on the bottom, the emerged lateral root density is also much higher than the controls. Here you see some more of the cell wall changes I was talking about. This is an SEM micrograph timeline of lateral root emergence from the earliest time point I could catch through a mature lateral root. And there are differences in the cell wall, including this separation event. So at this point, I'd like to conclude with our treatment system does synchronize and greatly improve the density of the emerged lateral roots. And it seems to really involve cell wall loosening, which could be very important for biofuels, because if we can understand this process, we can harvest this to cause the plants to harvest this natural pathway for the plants so that we can loosen their cell walls and get more sugars out of the cell walls. And we're going to go from here to harvesting this material to start identifying these pathways so that we can go further along. I'd like to thank EPSCoR for the funding and all of you for listening to me talk.